Hey everybody, my name is Alex Yin, and I am a second-year medical student at Virginia Tech Correllian School of Medicine. I work with Dr. Win Lagon in the non-invasive ultrasound lab at Virginia Tech, and I will be presenting some preliminary data of our study looking into low-intensity focused ultrasound to the insula, with both the attenuation of contact heat evoked potentials and reduced pain perception in humans. So starting with some background, the insula is found deep to the lateral fissure and covered by the frontal operculum and has both an anterior lobule and posterior lobule composing of individual gyri. The insula is a promising brain target for pain mitigation, as it not only receives the majority of the direct disynaptic spinal thalamic projection, which we know is a sensory tract that involves nociception, but also the contact heat evoked potential has been sourced to the insula, as we see in this figure, where the contralateral evoked potential tracings were recorded for various gyri of the insula. And there are other brain imaging studies that report insular activation during painful stimulus, with the anterior insular cortex mainly involved in the affective or emotional aspects of pain, while the posterior part of the insula is more implicated in the sensory aspect of pain processing. Now, the problem with trying to target the insula for neuromodulation is that it is deep within the lateral sulcus. In current non-invasive neuromodulation techniques, such as a transcranial magnetic stimulation, which we see in this figure, only have negligible portion of the electrical field actually reaching the insula, lacking both the depth penetration and spatial specificity that LIFU demonstrates, as we can see here, with a free water tank data overlaid to structural MRI with single gyro resolution. So with all this, we want to know if we can modulate the anterior and posterior insula with LIFU, and does it indeed affect neural pain signatures and the perception of pain? That leads us to our hypotheses. First, LIFU can target both the anterior and posterior insula individually. Second, LIFU to the left anterior insula attenuates the P1 amplitude of the contact heat evoked potential, or CHEP, affecting unpleasantness of the pain stimulus, while the left and posterior insula attenuates the N1 amplitude of the CHEP, affecting the intensity. And third, LIFU to the left anterior and posterior insula both reduce subjective pain perception. To test these, we utilize the quantitative sensory testing, or QST machine, as we see here, to provide a brief contact heat stimulus with a thermal probe placed on the dorsum of the hand, represented here and also here. And we use a perceived pain rating scale of 0 to 9. We began by thresholding each participant to pain of 6 out of 9. Then LIFU was stereotaxically targeted either to the anterior insula or posterior insula using the BrainSight neuronavigation system with a single element focused transducer with the following parameters. You can see the transducer here. We had an N of 16 for the anterior insula, or AI, and an N of 17 for the posterior insula, or PI. We counterbalance the inactive sham with LIFU to the left AI or PI, concurrently with EEG, using 40 stimuli with a random interstimulus interval, or ISI, of 10 to 20 seconds, and have participants rate their pain from 0 to 9. If we blow up one of the stims here, we can see that continuous EEG recording with CHEP stimulus delivered at time 0 and time locked to the LIFU stimulus with an outside of the skull intensity of 3.5 watts per centimeter squared ISPPA, starting 200 milliseconds before and ending 800 milliseconds after the CHEP stimulus that allows for that temporal overlap of the two signals. Our outcomes included both the N1 P1 peak-to-peak amplitude of the CHEP and the perceived pain rating. So we'll begin with the results of the anterior insula here. We co-registered the MRI and CT scans from each participant for modeling. And here's an example of one of the participants where you can see in the coronal cut, the insula here, with the transducer on the skull, and then the transverse cut here of the insula, and also the transducer here. And the various colors here represent the pressure in kilopascals. If we take a slice and view the insula laterally, we can see that peak pressure of about 180 kilopascals over the anterior insula, demonstrating that we indeed have single gyro resolution. For the EEG, we have that CZ electrode CHEP data, where the y-axis represents the amplitude in microvolts and x is time in milliseconds, with the red and black lines representing the average traces across those 40 trials for a single participant. The black line represents sham, and the red line represents LIFU. Below, we have that pseudo-color trial-by-trial map, where blue here represents negative and red is positive, all contributing to the average traces above. When you compare the maps between sham and LIFU, you can see more noticeable difference in the red here, or positive, representing that P1 amplitude compared to the blue here, or N1 amplitude. For our group analysis, the y-axis, there is the peak-to-peak amplitude in microvolts. We saw around an 8 microvolt reduction between the sham and LIFU, and this was statistically significant. For the pain perception data, the y-axis, there is the pain perception rating from 0 to 9, and we see a pain perception rating drop of about 0.5, which was not statistically significant. Moving on to the posterior insula, we again see the same modeling for one of the participants with a peak pressure of about 220 kilopascals over the posterior insula, where just like the anterior insula, we demonstrate single gyri resolution. We also saw an attenuation of CHEP peak-to-peak N1P1 amplitude for posterior insula, but compared to the anterior insula, we see a more noticeable change in both the red here and the blue in the pseudocolor maps for both the P1N1 between sham and LIFU, also demonstrated by the decreases in the amplitude here and the average tracing. We also can see a statistically significant 6 microvolts reduction between the sham and LIFU for the group analysis. And similar to the anterior insula, we did not see statistically significant differences in the pain perception, again with a drop in rating about 0.5. So for conclusions, single element 500 kilohertz transducer LIFU can successfully target both the anterior and posterior insula individually with single gyro resolution. LIFU to the anterior and posterior insula attenuates the N1P1 amplitude of the CHEP, and LIFU to the anterior and posterior insula decreases pain ratings to heat stimulus on the dorsum of the hand. For future goals, we would like to continue increasing the number of healthy normal participants to add to our current data to better compare and contrast AI versus PI effects on both the contact heat evoked potentials and behavior, while continuing demonstrating the neuromodulation abilities of LIFU. More importantly, we'd like to further test the relationship of the insula with pain to develop clinical therapies for chronic pain conditions, such as complex regional pain syndrome and fibromyalgia. Thank you to my lab family and mentors, the FBR SEAL Innovation for supporting our study, and also the Focus Ultrasound Foundation for hosting this conference. Thank you, everybody.